Hi everyone. Let us discuss about the DNA polymerase, basically which focus first on its structure. When we look into its structure, this DNA polymerase protein has three distinct domains. When we say domain in a protein, that means it is a independent part of a protein. That means so it forms a 3D structure on its own. That means so it folds on itself and forms a separate structural entity in a protein. So three such structural entities are seen in a DNA polymerase. That means three domains are present. These three domains are structurally distinct. These three domains, their functions are also different. So these three domains are they are independent structures of a protein. So what are these three domains that are present in DNA polymerase? So these three domains are finger domains, thumb domain, and palm domain. Three domains are there. So coming to their functions, so the thumb domain, so this helps DNA polymerase to tightly bind with the DNA. So once the polymerase loads onto the DNA, so this polymerase adds nucleotides before it dissociate from the DNA strand. So a single polymerase adds multiple number of nucleotides. Not like a polymerase can add only one nucleotide after binding to DNA and dissociates, and next polymerase binds and adds nucleotides. It's not like that. So that multiple number of nucleotides can be added by a polymerase enzyme. So adding of multiple number of nucleotides, we say it's a processivity. So processivity means how many number of nucleotides a polymerase can add to growing nucleotide strand in a minute. So that is called it's a processivity. Okay. So this thumb domain is very important for binding of the protein to the DNA, double-stranded DNA. Okay. So next one, fingers. So this fingers domain is also another important domain in polymerase enzyme. So where the nucleotides, so DNTPs, are added to the growing DNA strand. So that is a fingers domain function. The next one is that the palm domain, which is very important. So this palm domain has the active site. So in this active site, the nucleophilic reaction nucleophilic reaction takes place, in which the phosphodiester bond is created between the incoming nucleotide, and also between an incoming nucleotide and the 3' OH of the already existing nucleotide of the growing strand. So these three domains, they play a very vital role in synthesizing the growing DNA strand. So this is about the DNA polymerase enzyme. So this DNA polymerase enzyme has another activity, so which is not related to these three domains. So this activity is not related to finger domain, not related to thumb domain and the palm domain. So that is called exonuclease activity. So what is this exonuclease activity? When nucleotides are added to growing DNA strand, they sometimes a wrong nucleotide may be incorporated to the growing DNA strand. Wrong nucleotide means, so the DNA, the nucleotides, addition of nucleotides to growing strand depends on the sequence of the template strand. That means the nucleotides must be, the incorporating nucleotides must be complementary to the template DNA strand. Suppose adenine is present in the template strand, the thymine must be added to the growing DNA strand. So that complementarity must be present in the DNA synthesis. So when there is a mismatch between these two base, that means between the base of the growing strand and the template strand, so this nucleotide is removed by the polymerase itself. The nucleotide is removed by the polymerase itself. So for that, so the polymerase also possess nuclease activity, that is called exonuclease activity, so that the wrong nucleotide will be removed from the growing DNA strand. So the DNA polymerase synthesizes the DNA. That means it incorporates new nucleotides to the growing strand. Also, it removes if that nucleotide is not complementary to the template strand. So this mispairing of nucleotides can be corrected even after formation of the DNA strand also. So for example, while synthesizing DNA, the polymerase takes care of this proofreading. That means it ensures that there are no errors with respect to the complementarity. So this is during the synthesis of the DNA. So even after synthesis of DNA, some bases they may undergo some changes, base changes like mutations may happen in the DNA. So after synthesis of DNA, so to remove such a kind of mismatches between the daughter DNA strand and the parental DNA strand, so the cells possess a repair mechanism. So the repair mechanism performs after synthesis of DNA, and this proofreading mechanism performs while DNA is synthesized by the DNA polymerase itself. Okay, so this is about the DNA polymerase. Thank you.